Thank you so much for the invitation, kind introduction, and all the organizers of this wonderful conference. And originally, I'm from the Netherlands. I didn't show a map, but after studying in the University of Amsterdam, I moved to Berlin for my PhD. So a very brief introduction. Alcohol use disorder worldwide, 3 million deaths every year still result from harmful alcohol use. And in the United States, over 1 in 7 Americans are estimated to develop an alcohol use disorder in their life. And I just learned from Alexis that it's about 40% or so of Canadians drink more than 3 drinks per day, which I didn't look up the statistics, but that's high. However, fewer than 8% of adults who need treatment for alcohol use disorder actually receive treatment. Could be due to stigma, access to healthcare. But yeah, overall, there's a need for interventions that can promote long-term abstinence and reduce alcohol craving. And I actually came to the ketone field due to neuroimaging, not because of a nutritional background, but we see very clearly that if people drink alcohol acutely, there's a drastic decrease in brain glucose metabolism with about 20%. And this initially was being interpreted, you see how bad alcohol is, the brain doesn't function well. However, my previous mentor, Nora Volkov, used 2 tracers to study both glucose metabolism reduction, but also she saw a strong increase in acetate. So alcohol gets metabolized by the liver, gets converted into acetate. Acetate crosses over the blood-brain barrier and can be used by the brain as an alternative energy source. And during sobriety, we also see that heavy drinkers show higher acetate uptake. So the sobriety when people are not under the influence of alcohol use. So more acetate uptake in heavy drinkers compared to social or non-drinkers. And similarly, we see a longer reduction in brain glucose metabolism in people who drink alcohol regularly. So we thought, well, this is relevant for alcohol use disorder treatment. Namely, when people quit drinking and this source of acetate is not there anymore, it might be that during detoxification, this state of high acetate in the brain and in other cells of the body goes to low acetate, whereas glucose metabolism takes some time to recover, maybe up to 2 weeks. So it might be that just during prolonged detoxification, the lack of acetate may result in an energy deficient state and may contribute to withdrawal symptoms that we see during alcohol detoxification. And a ketogenic diet, I really don't have to introduce it here too much, but of course, it's a diet very high in fat and low in carbohydrates, and cells in the body, including neurons, could use ketones as a fuel rather than glucose, including acetoacetate, which is structurally very similar to acetate. So the goal of my lab right now is to really see, can ketone bodies, primarily acetoacetate, that is so similar to acetate, can they help overcome this hypothesized energy deficient state in alcohol use disorder? And when I started this project in humans, there was a lab in Denmark, in Copenhagen, from Anders Fink Jensen, that did his first study in rats. And he showed that alcohol-dependent rats who were fed a ketogenic diet during alcohol withdrawal had lower signs of alcohol withdrawal, namely lower body rigidity and irritability compared to those fed a standard diet. So based on that, we designed a first 3-week ketogenic trial, ketogenic diet trial in alcohol use disorder in patients during alcohol detoxification. And I found it extremely important for a diet intervention trial to make it blinded. So what we did is we gave 4-to-1 ketogenic diet or standard American 50% carb control diet in the shape of shakes. Namely, participants received for breakfast, lunch, and dinner a shake for their caloric needs that was either ketogenic or standard American. And on the side, they received some snacks that were all ketogenic. So in that way, people did not know which intervention they received. And also afterwards, we asked, which diet did you think you were on? And people did not know. Most of them actually thought they were on the ketogenic diet. But just as a proof of principle, we had an independent person tracking the acetoacetate in urine and then the BHB levels in blood. So you see that the ketogenic diet group went into ketosis and the control diet group did not. And I actually had a neurologist who treats people with epilepsy with a ketogenic diet look at this graph and he said, your patients went into ketosis so fast. But we couldn't really compare it because we didn't have a control group. But it's still another question. Could it be just what you said previously, lean versus obese people? Could it be that people with an alcohol use disorder, is it easier for them to be in ketosis? I don't know yet. But for us, the main clinical outcome was the need for benzodiazepine treatment during that first week of alcohol withdrawal when people really need this. So it was a psychiatrist who prescribed benzodiazepine medication for alcohol withdrawal. If alcohol withdrawal was high, people were prescribed benzodiazepines. And we saw a strong reduction in this need for benzodiazepine medication. And also the number of people who required benzo was reduced in the ketone group compared to the control group. We also did functional MRI. We looked at brain responses to alcohol cues and food cues and computed neurobiological craving signature. This was a signature that comprises the medial prefrontal cortex and the ventral striatum, so really brain areas that respond to reward and that were previously shown by Hedy Kober to be correlated with drug craving. And we saw that the ketogenic diet had lower neurobiological craving signatures compared to those on the standard American diet. And also behavior, people said that they wanted alcohol less, so it's a small factor. And then in a parallel rat study, we were wondering, does a ketogenic diet also affect alcohol consumption? We did this in collaboration with the National Institute on Drug Abuse in Baltimore. And so they basically trained Twister rats to self-administer alcohol. They were then on a ketogenic diet or a chow diet for 2 months. And after that, they were back on a regular chow diet and were being made either alcohol dependent through alcohol vaping rooms, or they were vaping air, so they were not alcohol dependent. And this graph is a bit too complicated, but basically what it shows is that the alcohol-dependent rats on a ketogenic diet were less likely to self-administer alcohol afterwards. So a history of a ketogenic diet reduced alcohol self-administration even 8 weeks after being exposed to a ketogenic diet. And also acutely, there's a lab in Spain that shows that when mice were on a ketogenic diet acutely, that they also self-administered alcohol less. So one of our hypotheses was that it's because of this difference in brain energetics, but we also saw a pharmacokinetic effect, and quite a dramatic one, namely that a ketogenic diet increased sensitivity to alcohol. So rats who were fed a ketogenic diet and were placed in these vaping rooms acutely while they were on the diet, they could not tolerate alcohol one bit. They had 5 times higher levels of alcohol in their blood compared to those on the standard chow diet. So this is usually why NIH doesn't fund a ketogenic diet trial in patients with alcohol use disorder, because potentially people overdose. If they drink a 6-pack of beer, then suddenly they drink 5 times more than that. But here, we still really need to understand what this means. This was a 14-hour overnight vaping of continuous alcohol. So that's a lot different than oral alcohol. And collaboration with NIDA, and particularly Leandro and Ruscolo, are still investigating why this is the case. So they dissected the livers of rats on a ketogenic diet, and they showed a downregulation of alcohol dehydrogenase enzymes. So it might be that a prolonged ketogenic diet exposure decreases this crucial enzyme that breaks down alcohol. So an interim summary, we performed the first ketogenic diet intervention in patients with alcohol use disorder. It was quite well tolerated. The diet reduced benzodiazepine intake and neurobiological alcohol craving signatures. And also in a rat model, the diet increased alcohol consumptions and increased alcohol sensitivity, potentially due to this alcohol dehydrogenase enzyme. And we're currently following up on that finding in humans. So we're putting healthy students on a ketogenic diet for 4 days and then bring them to the lab to see if this alters alcohol metabolism. But we already see that 4 days might not be enough to see changes. So I'll actually prolong the exposure. But there are some limitations of a ketogenic diet, particularly for this population, that may not be the most reliable in terms of retention or sticking to a diet. So the diet is rigid and it's difficult to maintain, and it has low retention rates. So particularly for people with alcohol use disorder, I'd be concerned if people really stick to the diet. And the exogenous ketone monoester, also called a ketogenic diet in a bottle, not quite, but it was developed by Richard Veach and Kieran Clark, as we've seen before. But this might be an interesting model to see also whether this has any influence on alcohol withdrawal and alcohol consumption. So the supplement, as we've seen before, reaches BHB concentrations in blood similar to BHB levels after a 2-3 ketogenic diet. And also, as we've seen before, is that Brianna Stubbs demonstrated that it's decreases appetite, so single dose. And in our first pilot study, now that we had 5 alcohol use disorder patients who came in for PET-FDG scan both after or at baseline and after a ketone supplement. Also, the ketone supplement decreased alcohol craving, and it increased, of course, blood BHB levels. And very similar to alcohol, ketone supplements also decrease brain glucose metabolism, or the metabolic rate of glucose, by 17%. So we scanned 5 patients with alcohol use disorder and 5 controls, and we were interested to see whether there were any differences in these reductions in glucose metabolism, but there were none. Basically, we saw very, very similar baseline levels of glucose metabolism, but also very similar reductions. And unfortunately, we couldn't do carbon 11 acetoacetate, but still, proof of principle, we tested the same dose of ketone supplements with protomagnetic resonance spectroscopy, and you see a clear increase in the detection of BHP in the anterior cingulate cortex. So obviously, the reductions in glucose metabolism were due to a shift from glucose to ketone to the brain energy source. And in a separate study, we really wanted to understand the alcohol pharmacokinetics and the subjective responses to alcohol after ketone supplementation. We changed a little bit from the delta G because it was really hard to perform the study with the delta G ketone ester. People really didn't like it. They were gagging. And then this brain kinetic came on the market and is really much better tolerated. So here we brought in 10 healthy volunteers. They came into the lab twice. Once they received a placebo with sugar, and once a ketone supplement, and then they received a low to moderate dose of alcohol. It's about 3 shots of vodka. And after that, we measured alcohol blood and breath levels and subjective responses. And we saw, there was a 30 minutes interval and people were fed. I didn't want to do this fasting because it's hard to metabolize alcohol when you fast. But we initially thought that the keto supplement, similar to the ketogenic diet, may actually increase blood and breath levels of alcohol. But to our huge surprise, we actually saw the opposite. So in red is plotted the breath alcohol and blood alcohol response after the ketone supplementation. It was very damped, and in some participants it was even 0. They were asking, did I really just drink alcohol? What was that? And also subjective responses after the ketone supplements were that they didn't really like it and they didn't really want more alcohol. So we saw that the ketone supplements strongly blunted alcohol breath and blood responses in healthy humans, unlike what we actually hypothesized. And in rats, Leandro just did this in a few days, even though for us it took several months to collect the human data. I'm always a little bit jealous of how quickly you can do animal studies. But we found very, very similar results, namely that the kinetic reduced blood alcohol levels in rats. So it's not a species difference. And also the kinetic has a sweetener allulose in it, which is quite a novel sweetener. So we were concerned that maybe that was driving this effect on alcohol pharmacokinetics, but clearly there was no effect of allulose. So the ketone supplement blunted alcohol blood responses in humans as well as rats. But then the question was, what is this effect? Is this a liver effect? Does alcohol get broken down faster? Or is it an absorption effect that it just doesn't get absorbed slower? And the answer was, it would have taken a lot of time to set up an IV alcohol lab, so we didn't do that, we bypassed it and just did this in rats. But we showed that an IP alcohol injection basically did not have an effect. So it has to be an effect on absorption. So it might be that ketones, particularly this product, delays the absorption of alcohol, possibly to delayed gastric emptying. And it's really interesting that some of the GLP-1 agonist work that we see in alcohol use disorder, it shows not an effect in a bar lab, not an effect on alcohol self-administration, but it does on blood alcohol levels and breath alcohol levels. So there might be a mechanism that is similar with GLP-1, or at least one of the mechanisms that are similar. So in conclusion, ketone supplement rapidly shifted glucose to ketones as a brain energy source, as shown with PET-FDG and MRS. And the ketone supplement delays alcohol absorption and decreased alcohol wanting and liking in healthy volunteers, and in a small sample also reduced alcohol craving in patients with alcohol use disorder. So ketones might be of therapeutic benefit to individuals with an AUD, but obviously much more research is needed. So for future directions, we need to find the optimal dose. Maybe this 4 millimolar is way too high, and we could actually also go to something like 1 millimolar. We don't quite know yet. And also we need to work on more palatable products, and preferably also something that is not so transient as a ketone supplement that just after 4 hours is out of your system again. And I'm currently working on the effects of ketone supplementation on alcohol consumption and on alcohol withdrawal. And we're measuring also brain energetic outcomes of mitochondrial health, including brain NAD levels. And ideally, acetoacetate, if we ever get it off the ground at Penn. And currently we have an ongoing study which is almost complete on the effects of ketone supplements and alcohol withdrawal. So we basically redid the experiments of the ketogenic diet, but then with ketone supplement drinks. So for breakfast, lunch, and dinner, people received just a regular diet plus 10 grams of ketones 4 or 5 days during alcohol withdrawal. And we currently randomized 48, and we'll go up to 50. So hopefully we'll get the first results soon. And then we have some preliminary data in rats that's both IP and oral administration of the ketone supplement reduced alcohol self-administration. But this was a really high dose. So ideally, we also look more at longer term intervention, maybe even interventions where the rats would self-administer, but they really don't like the keto supplement to just take it themselves. But it's promising, at least for human use. So many thanks to my little lab and also to Dr. Kinane, who hopefully set up the acetoacetates and FDG dual tracer technology at Penn, or at least with the alcohol use disorder. Yeah, for sure. Thank you.